Hello everybody, welcome back to another edition of Edpuzzle Lecture Notes. When we left off, we went over glycolysis and pyruvate conversion. Today we're going to go to the Krebs cycle, also called the citric acid cycle. So, like always, our daily objective, you want to identify what are the reactants and what are the products. So what are the things that go into the citric acid, or the Krebs cycle, and what are the things that come out? What are the things that get produced? So identify the reactants and products of the Krebs cycle, and it's also called the citric acid cycle. Alright, so this is where we left off last time. Remember, the first step of cellular respiration is a reaction called glycolysis. Glycolysis occurs within the cytoplasm of the cell. So it's just like the fluid in the cell that's not within an organelle. So it's not in the mitochondria yet. Now, glycolysis starts with that molecule we've been talking about for several weeks now called glucose, a 6-carbon sugar called glucose, C6H12O6. So in glycolysis, that glucose molecule is chopped right in half. So this is what we're doing. We're breaking down that glucose molecule in order to release energy. Now, to break that glucose in half, it takes 2 ATPs in order to snap it in two. And that's where it's going to eventually become 2 molecules of what's called pyruvate. Now, when that ATP is used to break glucose in half, you get some products. You get some energy out of it. And what you get is, you get 4 ATPs. Now remember, you really only get 2 because you had to spend 2 ATPs in order to break it, and then you earned 4. But since you spent 2 and earned 4, you really only got 2 ATPs net from that. And you also get 2 electron carriers NADH. So in glycolysis, it happens in the cytoplasm. The glucose sugar is broke into 2 pyruvates, so it's no longer C6H12O6. It is now 2 pyruvates of C3H4O3. Now, obviously, when you break the 6-carbon sugar in half, it makes sense that you get C3. It also makes sense that you get O3, because 6 oxygens split in half gives you 3 oxygens. Now, the reason why it goes from H12 in glucose all the way down to H4 is because first, we lose 2 hydrogens when we make NADH, and also 2 hydrogen ions are also released. So that's 4. So we lost 4 hydrogens in glucose, and that's why it went from H12 to H4. So all you're going to need to know here are the products of glycolysis. 4 ATP, but it's really only 2, remember, because we had to spend 2 ATP to make 4, so we really only got 2 ATP and 2 NADHs. Now for pyruvate conversion. Now in pyruvate conversion, we're going to get 2 more energy molecules of NADH. And this is also the time where 2 carbon dioxide molecules are released as a waste product. So you can see, when we lost these 2 CO2s, we lost 2 carbons, and that's why it went from C3 to C2, because we lost a carbon in each of those pyruvates. We also lost 2 oxygen molecules per pyruvates. That's why it went from O3 to O, because we lost 2 oxygens there when we made the CO2. So now we're left with C2H3O. You're not going to have to remember those chemical formulas, but just know the energy that gets released. By breaking down that glucose molecule, we keep releasing more energy. And the last product of pyruvate conversion, 2 molecules, 2 CoA enzyme molecules, 2 of them bond with each of these pyruvates to form the final product, which is going to go into the Krebs cycle, called acetyl-CoA. So you will need to know that. Basically, you need to know the reactants and the products of each of these steps. Alright, so here's the Krebs, also called the citric acid cycle. Now, this cycle, remember, glycolysis was totally anaerobic, did not require oxygen at all. Now, the Krebs cycle does not use oxygen, but it needs oxygen to be present in order for the Krebs cycle to work. So if there's no oxygen, the Krebs cycle will not work, even though oxygen is not actually used. Now, where in the cell does this occur? Same place as pyruvate conversion, in the mitochondria matrix. Mitochondria matrix, which is basically means the middle of the mitochondria. Now, what are the reactants? We need the two molecules that we made in pyruvate conversion. So we need those two acetyl-CoAs, those two acetyl-CoA molecules that we made in pyruvate conversion. And we also need dead batteries, but you really don't need to know this. We also need NAD, dead battery. It's going to come and pick up electrons. And we also need FAD, NAD and FADs. And those are also going to come and pick up electrons to be used in the electron transport chain. All right, so what are our products of the Krebs cycle? We get 2 ATPs. We're going to make 4 molecules of CO2. That's a waste. We're going to make 6 NADH electron carriers, so 6 electron carriers, NADH. And we're also going to make 2 of its cousins that also carry electrons, 2 FADH2. And all you need to know, those carry electrons to the electron transport chain. So we made 2 ATP in glycolysis, 2 net ATP, and we got 2 more here. So we're up to 4 ATPs on our way towards 32. All right, so here's a little diagram of the Krebs cycle. So here's one of the 2 acetyl-CoA molecules that were made in pyruvate conversion. So remember, we started with glucose, we broke it in half into 2 pyruvates. Then we broke it down further in pyruvate conversion to turn it into acetyl-CoA. Well, it comes in to the Krebs or citric acid cycle, and you can see it's C2H3O, and it's going to bond with a molecule called oxaloacetate. So the C2H3O bonds with the C4H2O5. You will not have to know that, so don't freak out. And it becomes a molecule called citrate or citric acid. That's why it's also called the citric acid cycle, C6H5O7. Now, when it goes between citrate and what's called alpha-ketoglutarate, this is where we start getting some energy. This is where the first carbon dioxide is released. That's why you can see it goes from C6 to C5, because we lost 1 carbon in the carbon dioxide. It also goes from O7 to O5 because we lost 2 oxygens when that CO2 was given off. We also make 1 electron carrier, NADH, and that's why it went from H5 to H4, because we lost an H right there to the NADH. All right, so now when it goes from ketoglutarate to succinate, in this reaction we're going to get 3 products. Remember, all we're concerned about are the products. Here we're going to get another CO2. We're going to get another of the electron carriers, NADH, and we're also going to get 1 ATP from this. So far we got 2 molecules of CO2, 2 molecules of NADH, and 1 ATP. The next one, remember, you're not going to need to know the names of these molecules or their chemical formulas, anything like that. You just need to know the products. So when it goes from succinate to fumarate, this is where we get NADH's cousin, FADH2. And it's also an electron carrier. You can see it went from H4 to H2 because it stole 2 of the hydrogens. But more importantly, it picked up electrons to carry to the electron transport chain. Now between fumarate and malate, nothing really happens other than water is added to it, but that doesn't really matter. And when it goes from malate back to oxaloacetate, we get the final NADH, the final electron carrier NADH. Remember, that's for the first acetyl-CoA. Remember, with glucose, we make 2 of them. So it's going to go around again with the second acetyl-CoA, and you're going to get these same products again. You're going to get 2 more CO2s to make 4. You're going to get 3 more NADHs for a total of 6. And you're going to get another ATP for a total of 2. And also another FADH2 for a total of 2 also. So that is an overview of the Krebs or citric acid cycle. But remember, don't think this is that hard, because all we're concerned about are the products. what is produced during this cycle. We're not concerned about the chemical formulas or the names of the molecules. All right, so let's go ahead and stop and break it down. Let's see what these products are. So in glycolysis, we made a net of 2 ATPs. Remember, we made 4, but we also had to spend 2 in order to break glucose in half. So we really only made 2. We got 2 NADHs from glycolysis. We didn't get any FADH2s, and we got 0 CO2s. Now in pyruvate conversion, we got no ATPs. We got 2 NADHs. We got no FADH2s, and we got 2 CO2s, 2 carbon dioxides. Now in the Krebs or citric acid cycle, we made 2 more ATPs. We made 6 total NADHs. We made 2 FADH2s, and going around twice with both acetyl-CoAs, we made 4 carbon dioxide molecules. So we're done with all of this stuff. We're not going to make any more NADH, any more FADH2s, or any more CO2s. We do have a lot more. We have 28 ATPs to be made in the electron transport chain, and we still need to make our another final product of 6 H2Os or 6 water molecules that will also be produced in the electron transport chain. All right, I hope you enjoyed it and understood a little bit of it, and I'll talk to you next time. And for those of you guys that took notes, here's a real easy little review, little quiz. For those of you guys that didn't take notes, good luck.